Welcome to a new chapter of glioblastoma, aka GBM, presented by the Glioblastoma Research Organization. This season, we're adopting a documentary-style format to offer you a never-before-seen look into the world of glioblastoma from around the globe. Join us as we venture into labs and companies worldwide, providing a comprehensive view of some of the cutting-edge efforts to combat GBM. Through intimate interviews and detailed on-site explorations, we'll offer an insider's perspective on the innovative strategies and real-time progress in the fight against glioblastoma. Thank you to our sponsors whose generous support has made this season possible. Please note that the sponsorship of this season does not constitute an endorsement of any sponsor's products or services. Remember, the information presented is for educational purposes only and it is not intended to treat, diagnose, or prevent any disease. Always consult your medical provider and healthcare team with any questions.
Hi, my name is Javad Nazarian. I am a translational scientist. I am the head of the DMG research program in Zurich, Switzerland, and an adjunct professor at the Translational and the George Washington University in the US. My background is in human genetics. From early on, when I developed my career, I have focused on pediatric brain tumors. When we started studying pediatric brain tumors for the high-grade gliomas, specifically for midline gliomas, there wasn't much known about the genetic profile of these tumors. We didn't know much about the tumor biology. Within the past decade, we have been able to learn more about the biology of these tumors. In that context, my team tries to develop drug and drug therapies that can target this genomic perturbation in this particular patient population. The main effort in our lab includes working very closely with the clinicians. I believe in the power of the translational medicine. And the translational medicine is to be able to rapidly translate the findings that we have in the laboratory into the clinical setting. For the past 50 years, we've had a number of different clinical trials that have really run based on minimal amount of knowledge of the tumor biology. In the past decade, we've been able to decipher the genomic profile of these patients. We know more about what pathways, what molecular perturbations drive the tumor. The mission of our team is to use this data to combine therapies that are either novel drugs or repurposed drugs, to combine them to develop therapies that can effectively reduce the tumor size in these patients, and ultimately to find a cure. My personal connection to the field started with a family who had lost a child with brain tumor. The family contacted me and asked if we could develop a program that could be studying pediatric high-grade gliomas. This is when we had no knowledge, zero knowledge of the biology of these tumors. And with the help of the family who funded us back then, we developed a small laboratory that now has grown into an international effort from Africa to Europe to the United States that focuses on pediatric high-grade gliomas. We believe in the power of international collaboration for rare diseases, for pediatric diseases, for pediatric brain tumors that are really challenging. I think most of us believe that the power of international collaboration can push the field forward faster. Having access to tissue specimens, developing the cell lines from these patients, sharing data, sharing drugs, sharing knowledge is really important in this particular field, specifically given that the number of patients are not as high as what we have for some of the adult entities. And for pediatric brain tumors in general that are really challenging, the collaboration, international collaboration is really important. The main theme and the main power behind this is that brain tumors and cancers don't have religion, they don't have boundaries, they don't have nationalities. They happen everywhere, at every corner of the world. And we've been able to develop a platform where we share data readily within the labs, a consortium that we have developed that again includes Australia, Europe, Africa, and the United States to share data, but also share resources readily. We try to overcome some of the limitations that we have for transfer of data, for transfer of specimens, for the sheer factor of pushing the field forward as fast as we can. We've had a huge progress in understanding the biology of these tumors. 10 years ago, we had almost zero knowledge of what is driving these tumors. We had no distinction between pediatric and adult tumors. Brain tumors were just brain tumors. Within the past 10 years, we've been able to assign zip codes, assign entities to these tumors. Some of the particular brain tumors have multiple subtypes. We've learned that not every tumor is exactly the same as the next. We have subtypes, and the subtypes really allows us to have personalized and precision therapies and precision medicine. We know now that some of the tumors that arise in younger children are very different of the same tumor, same location that arises in the older patients, and the same as those tumors in adults. The brain tumors, they do share pathways, they do share molecular entities, but they're different. So we have learned a lot. We have learned a lot to push the field forward. Some of the challenges that we have include getting the drugs to the tumor. We need to have entities and platforms that can get access to the tumor. Our team has been very helpful and successful in developing some of the drugs that can get into the tumor and be activated in the context of focused ultrasound, for example, which is a technology that I believe that will revolutionize the way that we get into the tumors, the way that we expose the drugs into the tumors, which currently remains as one of the main challenges for solving any of the brain tumors, including adult and pediatrics. One of the main challenges that we have in brain tumors is the lack of visibility. For most of other tumors, a surgeon can always go in, obtain a tissue specimen, study this in the context of RNA, genomic, proteomics, and then devise a plan for the treatment of that patient. And then the surgeon can go back in and take tissue again to see how that tumor really changed. For brain tumors, we don't have this visibility. We don't have this accessibility to go into the tumor and take the samples out, to sample the tumor longitudinally. For that reason, we have a lack of clarity of how the tumor evolves and changes in the context of the treatment. We had proposed a study to the Glioblastoma Research Organization to fund us to have a platform that we could take longitudinal biopsy of an in vivo model that we are studying in the laboratory. Longitudinal biopsy is where we treat our model that we have in the laboratory, and we go back into the model. We take a biopsy, which is a very limited surgical study that we take a small sample from the tumor out, and we can study this. This longitudinal sampling and longitudinal biopsy allows us to look at the tumor. And it's really cool because you can treat the tumor, go back into your model, take a biopsy, and do molecular profiling to see how the tumor change. Because we believe that most of the treatments that we have in the clinic, they do work, but the tumors are fairly smart. They do change their pathways, they compensate for some of the pathways that we are targeting, they activate alternative pathways. But to us, we are absolutely, we have a lack of knowledge of how the tumor is evolving and how it's changing. Taking biopsies in the context of time, longitudinal, and in the context of treatment, teaches us how the tumor is changing. And in that context, we can devise the next level of treatment that then can target those compensatory pathways. This has been done for other diseases. For leukemia, for example, it's very easy or much easier to access a longitudinal sample from the patient to see how they're responding. For our brain tumor patients, this is not possible. The funding that we have received allow us to do this, to ask such questions, treat our in vivo models that we have with a particular treatment that is clinically relevant, to go back to the model, biopsy, perform profiling, and see how the tumor change, identify those compensatory pathways, and to target them again. This data can be readily then translated into the clinic because, again, we firmly believe that these tumors are targetable. We can target them, but so long as we have a clarity on how the tumor is responding to the treatments that we are devising. We completely understand that for patients who are diagnosed with brain tumors today, this is a big challenge, and many parents and children and patients ask the question of what are the advancements in the field. We know that in the past 10 years, the past decade, we've been really able to define the molecular biology of many of these tumors. We know so much more today than we knew 10 years ago. Most of the progress that we have done in the past 10 years has been supported by families, has been supported by patients who've given us tumor specimens to study. Because of these donations of the specimens, biospecimens, tumor samples, blood, CSF, because of the financial support of the families that have been able to support our research, we have pushed the field forward. There are platforms that are developing now that are emerging that are being used in the clinic, including focused ultrasound, where non-invasive ultrasound opens the blood-brain barrier, which allows for the passage of drug into the tumor, which has been a huge problem for translation of the preclinical data to the clinic. We have spent millions of dollars to define drugs that can target the tumor in the laboratory, but we have failed to get those drugs and drug combinations to the clinic. And I think one of the major problems has been drug-tumor exposure. We just have not had that exposure. I think that focused ultrasound will get us that opportunity when we can get the drugs into the tumor bed. Another emerging platform for brain tumors is the immunotherapy. We know that for leukemia, for blood tumors, we have been able to cure some of these patients using immunotherapy. For brain tumors, that's a challenge because we don't really know what is the activity level of immune system within the brain. There are modalities, there are platforms that are being developed now that can reactivate that immune system that we have within the brain tumor, which is typically a cold immune platform. We know that some of the drugs that we give to these patients reactivates the immune system within the tumor bed in some of these patients. We have seen this, where we take the rebiopsy, or unfortunately, when we look at the autopsy from some of our patients, we do see this has been reactivation of immune system that has been caused by the drugs that we have been giving to the patients. A combination of such drugs that reactivates the immune system plus the emerging immunotherapies, I think, would help to increase the tumor response and reduce the tumor growth. A third avenue that is growing fast and is helping pediatric brain tumors, helping many of the tumors across the board, is the AI. We have developed platforms that can take a specimen from a patient and can detect the presence of different antigens, which are different proteins that are within the tumor, just using AI. And the advantage of this is that for brain tumors, we don't have so much time. When a patient is diagnosed, the tumor is taken from patient. Every minute, every hour is count. The sooner that information about the tumor, about the biology of a tumor can be disseminated and sent to the clinic, the faster clinic can come up with a treatment modality for that patient. AI and digital imaging is actually called, of a tumor specimen can allow us now quickly detect tens of antigens, of antibodies that are present within the tumor of a patient. That data can go to the clinic. If the clinic wants to implement an immunotherapy for that patient, those data could be very helpful for them. This rapid identification of protein RNA expression within the tumor is something that could help the clinic a huge amount, because currently, the same analysis will take days, if not months, to be analyzed. So to shrink the time of diagnosis of the tumor type and type of specimens that we have will help to guide the clinic much better and faster to advance the therapy or precision therapy of their patients. We hope that collectively, the combination of emerging AI platforms, of immunotherapy, of focused ultrasound, of more drugs that are more clinically valuable and relevant to this patient population, a combination of these treatments would allow us to treat some of the patients and effectively treat most of the patients that are being diagnosed with brain tumors, which currently stays a challenge for us. I think collectively, clinical studies have pushed the field forward. A decade ago, we didn't know anything about the biology of these tumors. Within the past decade, we've been able to push the field forward. Clinical trials are much smarter now. They're more personalized. They're more dedicated to the patients. Neurosurgeons take a biopsy from these patients to study the tumor biology, and that is used to implement a therapy that is effective for that patient. I firmly believe that the collection of the emerging technologies that we have will definitely find effective therapies for patients who are diagnosed with brain tumors.
Hello, my name is Kamil. I'm a postdoc in Javad Nazarian's lab, and my main interest is pediatric gliomas. I was doing my PhD in adult glioblastomas, so that brought me to the field of pediatric ones as a change the topic, and then hopefully, so our research will be beneficial for our little cancer patients. I'm working currently on FOXO3 transcriptional factor, and then we're trying to identify this factor as a promising target in the DIPG research.
Hi, so I'm Matteo Bimbati. I'm a new PhD student in Professor Javad Nazarian's lab. Actually, I just started, I studied my PhD last July, and I joined this lab working on the CRISPR and DMG. So during my PhD project, I'm trying to investigate DMG vulnerabilities using the novel CRISPR-Cas9 technology. So I'm performing two different arrayed CRISPR screens targeting transcription factor and RTKs.
Hi, my name is Bettina Kritzer. I'm a research associate in the research lab of Javad Nazarian in the University Children's Hospital in Zurich. I work mainly in the lab. So I process patient samples and I do research on them. And I also do some project management and coordination with collaborators. Because of our research field, we collaborate with many different organizations to also move the field forward. So I wanted to work in the brain tumor field because I had a childhood friend that passed away because of a high-grade glioma. So I studied biotechnology. And I wanted to go into research, not specifically brain tumor. So one project I'm mainly working on is when we get fresh patient tissue from biopsies. So a part of the biopsy is always getting for the diagnosis, but when we're lucky, we also get a part for research. So we process this tissue and we're now developing a platform to rapidly screen a lot of drugs on them to then within few days, being able to inform the clinic about our results and maybe possible treatments for the patients. And in addition to that, I also work on different collaborations where we incorporate immunotherapy, focused ultrasound, or also AI platforms to improve our research. So for me, typically in the morning, I start first with going to the lab, start to look on all the cells that we have in culture, if everything is fine. So we plan experiments, we have some meetings with collaborators, also team meetings where we look at different experiments, maybe hurdles we have to tackle so that we work together. So I think one of the biggest hurdles in our pediatric brain tumor area is just that it's a rare disease. We don't have that many patients. So it's super nice that we actually work globally together to also collect data and specimens from all over the world. Also, we have hurdles in the platforms we have at the moment. So what we use in the lab, we know some of them are maybe a bit outdated. So we test the drugs in our in vitro setting, and now as we know that it's not that translatable, we actually work on better platform. We're developing new methods that this translation to the patient will be better.
Hello everybody, my name is Michael Grotzer. I'm a pediatrician, pediatric oncologist and specialized for pediatric neuro-oncology. Currently, since 2018, I'm the medical director and the chair of pediatrics here at Children's Hospital in Zurich. I'm glad to have you in this audience. We just moved to a new Children's Hospital in November, and I'm extremely proud of having the research facility and the hospital in close neighborhoods. So that's the way we want to push medicine into a new direction. So whatever we have in terms of problems on the clinical side, We transport it to the research groups in order to make novel treatments and novel ways of better diagnosis possible. And with this close relationship, truly convinced that here in Zurich, with the new University Children's Hospital, with our fantastic research groups, we are able to make a step into the right direction with significance, not just for Zurich and for Switzerland, but for Europe and globally.
My name is Sandra Dias. I'm a consultant neurosurgeon working at the University Children's Hospital of Zurich. The reason why I became a neurosurgeon 20 years ago, that was when I decided to pursue this career, was because my brother was diagnosed with a brain tumor. And from that moment on, I knew that I would want to dedicate my whole life to the diagnosis and treatment of patients harboring brain tumors, because I know how devastating that can be to a family. And especially with the high-grade tumors, patients and families, they hold on to every hope and weapons they can find to try to fight this disease, even though we know that in a lot of cases it's a battle that we are having, but we cannot win. And this is my biggest motivation to work as a neurosurgeon and to make part of the research team, because I believe that, as mentioned before, during the last couple of years, so many improvements and new findings, new discoveries have been done, that we are really on the right way to find more personalized treatment and maybe one day to find a possible cure for each patient that we are working in. Parallel to this family experience that I've been having, there is also a patient that was diagnosed 4 years ago with a high-grade tumor. At that point was called the glioblastoma. Now with the new classification, the nomenclature has changed, but that is not the main point. So at that time, he was still a child. He was 17 when we operated him. And I've been quite intensively also living the hopes and the struggles of him and of his family, with the tumor responding to therapy, but then we see the recurrence again, and then they start the battle once again. And this is apart from the fact that we are still taking care of the patient here at the Children's Hospital. And the interesting about this case was that 4 years ago, when we sent the tumor to the lab, it was not just analyzed to know what type of tumor it is, but one part of the tumor was also sent to the lab of Professor Nazarian. And to the panel drugs that the tumor was submitted, there was a specific drug that was developed at the lab. And when this patient received the treatment, the tumor significantly regressed. And I think for a glioblastoma patient, being alive 4 years after the diagnosis in a very good health condition is something admirable. And of course, it's not just the therapy and the research, but it's, and that I can confirm, it's mostly the family and the support, the belief that the people have that is going to be cured. Because even though now the tumor has spread, he is still in an amazing condition, being able to meet his friends, to spend time with his beloved ones. But of course, that is only possible due to the background work that we as a team, neurosurgeons, the nurses, the neuro-oncologists, the researchers, that we all work together so that these patients can live longer and with a better quality of life. And of course also the neuroradiologists and the colleagues that are working with us doing the proton therapy, they also play a very important role. Another project that we initiated at our institution, parallel to the AI diagnosis of the brain tumors, is the implementation of a software and hardware that has been developed. It's called the NIO, in which we can make the diagnosis of a tumor sample intraoperatively, and based on that, probably being able to start the treatment immediately, which might help to achieve a better control of disease. But not only that, when we then extrapolate the use of this hardware into the research setting, maybe in a short future, we will be able to test some drugs in the tumor sample and then observe in vivo, in a reality setting, how the tumor is responding to the treatment. And based on these fast advances that we are fortunately experiencing and seeing in the clinical and research setting, when a patient and the family comes to our department with a diagnosis of a brain tumor, particularly a high-grade tumor, I always tell them one side, not to lose their hope, because no one knows what tomorrow will bring us. We have been having the testimony of such a fast growth of knowledge that I'm pretty sure it will even grow faster the next couple of years. And maybe the day where we will be able to cure these patients or to achieve a radical remission of this disease is shorter. The day is maybe closer than what we can expect. So as I always say, continue to live your life as if nothing was in it. Only think about the disease when you come to the appointments, because unfortunately we still have to face the reality and we know they are there. But live the life the better you can. Enjoy the moments. I mean, all of us should be doing that, because no one knows what tomorrow brings, and maybe it will be a good surprise.